Good morning, students. I hope you all are fine and I wish you have gone through what we discussed yesterday. But till now, we have discussed about the characteristics and the functions of the plasma membrane. Moreover, we discussed about how the movement of substances occurs across the plasma membrane or across a particular cell. That is by the process of diffusion and by the process of osmosis. One more characteristic of the plasma membrane which we discussed yesterday was that plasma membrane is flexible. On the basis of that flexibility of plasma membrane, I want to introduce you about a term which is called as endocytosis, and it is also known as phagocytosis. The flexibility of the plasma membrane, it enables the cell, it allows the cell to engulf the food from the external environment. This process of engulfing the food by the flexible movement of plasma membrane is called endocytosis. For example, amoeba. The amoeba is a unicellular organism and its cell membrane is quite flexible, so it is capable of extending its cell membrane around the food particle and can engulf the food particle. So this process is called as endocytosis. Now Vijay, the outermost covering of all the cells is the cell membrane, but in plant cell there is one more rigid covering outside the cell membrane which is called as the cell wall. This cell wall is a rigid layer and it is made up of a complex substance called as cellulose. In plants it is made up of cellulose. In fungi, this cell wall is made up of chitin, and in bacteria, this cell wall is made up of murein. These cellulose, chitin and murein are the complex substances. Complex substances, why we call them complex substances? Because they are formed by the joining of many simple substances, simple sugars. What are the functions of these cell membrane? First, the most important function, since it is quite rigid layer, it provides a fixed shape to the plant cells and provides the mechanical strength to the cells. Mechanical strength means allows the cell to withstand the external conditions. Then it also helps the cells to withstand very dilute external media without bursting. When we put, as we discussed, we put a cell in the hypotonic or the hypertonic solution. The hypotonic solution is extreme dilute solution, and in that case when the cell gains water, it may gain excess of water and it may burst. So the cell wall, it prevents the cell from bursting. It protects the cell from mechanical injury as well as from the entry of pathogens. It does not allow the pathogens to attack the cell. So these are the characteristics and the functions of the cell wall. Now Bache, when we put a plant cell in a hypertonic solution, then what happens is it loses water through osmosis. We have already discussed. So there is a shrinkage of cell contents. Shrinkage of cell contents, and this phenomena is called as plasmolysis. You see, this is the normal plant cell. This outermost layer is the cell wall. This is the cell membrane, and we know in a plant cell there is a large vacuole and it is the nucleus which is shifted to the periphery. Now when this normal plant cell is kept in hypertonic medium, that is a strong solution of sugar or salt, then the contents of the cell, they shrink because they lose water, and this phenomena is called as plasmolysis, and this cell is known as a plasmolysed cell. Now again, when this plasmolysed cell is kept in a hypotonic medium, that is in a pure water or in a dilute solution, it gains water through osmosis and the cell swells up and retains its original shape. The cell membrane again goes near the cell wall. You see here the cell membrane has moved away from the cell wall. Here again it has come near the cell wall. So in a hypotonic solution, when we put a plasmolysed cell in hypotonic solution, it gains water and swells up. Such a cell is called as deplasmolysed cell, and the process is called as deplasmolysis, that is the reversal of plasmolysis. Then later in the chapter you will find an activity which will show the significance of plasmolysis. Now this process of plasmolysis, it demonstrates the permeability of cell wall, as well as it helps in identifying whether a particular cell is living or non-living. Because remember beta, plasmolysis, or we can say the osmosis or loss or gain of the water, always occur in the living cell. It never occurs in the dead cell. Related to this, there is one activity 5.6 in your NCERT book. What is that activity? In that activity, two setups have been taken. Firstly, a Rio leaf a peel has been removed from the Rio. Rio is the name of a plant, has been removed, and it is examined under the microscope. When it is examined under the microscope, you will find the clear structures called as chloroplasts in that. When on this peel, a strong solution of sugar and salt was added and it was again observed under microscope, it was found that the contents of cell have shrinked, because the strong sugar or salt solution is a hypertonic solution and the plasmolysis occurs. But in the setup B, that Rio leaf is firstly boiled. On boiling, the cells will become dead. When the cells are dead and when that leaf is mounted on a slide and a strong solution of sugar or salt is put over that, then there is no change. Why there is no change? Because no osmosis has taken place. There is no loss of water. So it proves, this activity demonstrates that osmosis does not occur in a dead cell. So there is no plasmolysis. This is the significance of plasmolysis. Now one most important question which comes into our mind is, why a plant cell does not burst when kept in a hypotonic external medium? When we have kept plant even in pure water or in any dilute solution, why doesn't it get burst? In the functions of cell wall, I told you that the cell wall protects or helps the cells to withstand dilute external medium. Here comes the reason. When we put plant cell in a hypotonic medium, then it swells up. When the cell swells up, the cell membrane builds up a pressure against the cell wall. You see, the cell membrane is putting pressure on the cell wall, and this pressure is called as Turgor's pressure. When this Turgor pressure is built up, then there is one more rigid layer outside the plasma membrane, which is the cell wall. Cell wall exerts an equal pressure towards the cell membrane. So this wall is exerting an equal pressure. This is called as wall pressure. So Turgor pressure is the pressure exerted by the cell membrane towards the wall. Wall pressure is the pressure exerted by the cell wall towards the plasma membrane, and Turgor pressure is always equal to wall pressure, which does not allow the cell to burst. That is why In a plant cell there are lesser, because plants have to face more diverse conditions, environmental conditions, extreme conditions as compared to animal cells. In animal cells, since there is no cell wall, there are always the chances that cell may burst in the extreme hypotonic mediums. So this is all about today. Thank you.